My name is Abigail Lammers, and I'm a senior at Trinity. My presentation is called Shaping the Bacterial World, Bacteriophage in Chicagoland Waterways. So what is this project? First of all, this project is part of the bigger Urban Waterways project on Trinity's campus, where there are different groups of people researching different areas of the waterways around campus. My particular project looks at bacteriophage, which are small viruses that infect bacteria in water or in soil or just in the environment in general. What the goal of this research project is to really look at whether or not we can shape or change bacterial populations just using phage. Why would we want to do this? The reason that we would want to do this is because this is a more natural way of cleaning water, because we can use natural phage counterparts to infect their bacterial counterparts in water and clean it up. So in past semesters, what this project has done, we've done a metagenomic analysis of the Cal-Sag, Navajo, and Tinley Creek. This basically gives us a breakdown of all the bacteria that we're able to find in the waterways to tell us basically what we're working with. And then through other experiments, we determined a focus bacteria called Kaoxitoka, which is a human pathogen that we found in the Tinley Creek in pretty large quantities, actually. So we've been focusing on that one specifically. We've also done smaller scale experiments where we have actually shaped bacterial populations in very small amounts of broth, usually around 100 microliters. So these are really small samples that we've done. This year, what we were doing is what I like to call the water experiment, which is a large-scale bacterial experiment where we take a half a liter or 500 milliliters of water from the Tinley Creek and then put bacteriophage into it to try to shape the population of bacteria. And then we sent these bacterial strains off to Mr. DNA in Texas to have them sequence it for us. And then we also made a wild Kaoxitoka phage stock. We did this by taking wild Kaoxitoka phage from the Tinley Creek and then amplifying it a lot. And now we have a pure stock of that. And then we also sent that off to Mr. DNA to be sequenced. So why does this matter? This matters because the end goal of this project has always been to improve public waterway health, because this is a more natural way of cleaning up water, like I said before, because it doesn't use chemicals, it uses natural bacteriophage counterparts. We also did the water experiment because it mimics a real ecosystem, because instead of just having broth inside of a tiny little tube, we're actually using almost a liter of water, and the water is natural, it's straight from the creek, so we can see if we're actually shaping that wild population of phage. It also makes kind of a complete story for us because we've been working with these waterways for about 4 years now, so this would kind of give us a chance to see what the differences we've made are and kind of all the information that we found. So our next steps, when the data comes in, the data has been sent off to Mr. DNA, but in all the craziness of this pandemic, it hasn't come back yet. So when that comes back, we're going to quantify it and figure out if the water experiment worked, if we actually shaped those bacterial numbers, and how much we impacted those numbers. We also would really like to use Argon's electron microscope to get a picture of our phage. This is just a picture I got off the internet, but we want to get a picture of our phage to know what we've been working with for the past several years. And then after that, we want to compile all of it into a research paper and hopefully get it published in a waterway journal. Thank you for listening to this presentation.